closer look at those podocytes and talk about glomerular filtration. We're going to look at what is what I call filtration barriers. And I shouldn't say I, lots of people call them filtration barriers. It's not exactly like I coined the term. Filtration barriers refer to the barriers that fluid must pass before it enters into the renal tubules, the lumen of the renal tubules. These filtration barriers, if we zoom in a little bit and take a closer look at this here, the filtration barriers include first the capillaries, which the capillaries of the Bowman's capsule, the capillaries in the renal system, are fenestrated. So here I'm coloring in an epithelial, simple squamous epithelial cell. Color these in here. And you can see these simple squamous epithelial cells, but you can also see these large gaps that exist between these simple squamous cells. And those large gaps are called fenestrations, which means window. But they're larger than you typically find in capillaries, and that's because it's to our advantage to move larger volumes of fluid than we would in capillaries elsewhere. Remember the cardiovascular system, that the capillaries do in fact filter, right? You might remember net filtration rate versus net absorption. In the capillaries, as we talked about the circulatory system and capillary function, so substances, fluid, and soluble molecules filter out of the capillaries into the interstitial fluid, and then substances are absorbed from the interstitial fluid back. Fluid and other dissolved substances are absorbed from the interstitial fluid back into the capillaries. That's how capillary exchange works. But here we filter a much larger volume because of these fenestrations, and so a large volume of material will filter in through the capillaries, and you can see that shown here with this red arrow. This substance is being filtered through, but the capillaries are not the only barrier that needs to be crossed. This interstitial fluid, shown in blue, is filled with connective tissue, and this loose complex of collagen fibers, okay, and it's very loose. This would be lots and lots of room for fluid to move, act as a net or a sieve to catch larger cells and other structures like that. And then we have the foot processes. These are part of the podocytes. Let's go back over here and look at the podocytes here. The podocytes are these really cool little cells that are part of the Bowman's capsule, and these specialized cells, podocyte means foot. Well, podo means foot, and cyte means cell. And in this case, these are cells with many feet, you can see, and they extend their little fingers. I like to think of them as fingers. And these little fingers make what we call filtration slits. Okay, you can see those here. And so those filtration slits, going back to what we were looking at previously, this would be one of those little fingers, just a cross-sectional area of those fingers connected to the podocyte. Okay, and those podocyte fingers, if you take a look at your hand and you spread your fingers out, there's a lot of space in between your fingers for substances to move, and you're spreading it out in the air, but the air is going to move through. If you were in water and you were wiggling your hand in the water would go in between your fingers. But then if you close your hand so that your fingers are all touching to one another, some substances will get in between the spaces, but not as much. And even when you swim, I've been trying to teach my little 5-year-old to swim, and she has a hard time remembering to keep her fingers closed, but you need to keep the fingers closed so that you can push more water and propel yourself forward that way. And so, less water gets through those fingers if they're closed tight together and there's no space in between each digit. Filtration slits work similarly. The little toes can be spread apart and nice and wide, in which case the filtration slits are very large, and fluid will filter into the Bowman's capsule relatively easily. Or you can close those down to reduce the amount of filtration into the Bowman's capsule. And so this actually does occur to help control the rate at which fluid moves in. and how much fluid moves in. The red blood cells that are present in the capillaries are just too darn big. They're not going to get into the this is the yellow part here is the lumen. Okay, the lumen. Oh look, it's even written right there, so I just, took up extra time writing lumen when I didn't need to. Anywho, red blood cells are too big. They're not going to get into the lumen of the Bowman's capsule. Likewise, white blood cells are too big. They're not going to get into the Bowman's capsules. It's all sparkly. How fun. Okay, white blood cells too big, not going to get into it. Plasma proteins also too big. And so we'll just kind of draw some of these. Most of the plasma proteins, I should say. Every now and then a little bit of albumin will sneak in. Okay, but in general, they're too large. They aren't going to get through. If you have damage, so there's actually a number of different ways to open these podocytes up. If we have an infection, let's say we have a renal system infection, so our kidney infection. Usually those infections remain in the urethra, but not always. And so let's say we've got some bacteria swimming around in here and causing a whole bunch of problems. Well, that's going to activate the immune system. We're going to get histamine being released, and that's going to open up the capillaries, and it's also going to open up those podocytes. And at that point, these little white blood cells can be like, woohoo, and get in. Okay? And if the white blood cells can get in, the red blood cells can get in too. Boop, boop, boop. Because remember, red blood cells are actually smaller than the white blood cells. Not that I drew that accurately, but recall that that is true. And so one of the characteristics of a renal or urinary system infection, a UTI, is you might have red blood cells in the urine. You also might have white blood cells in the urine. And so that essentially means you've got a kidney infection that occurs. You're also going to get some nitrates and other substances in there that are not supposed to be there. And so those are things that are kept track of and measured by a urinalysis test. Lucky you do not have to pee in a cup. If you were taking this class during a normal, reasonable semester instead of the world of pandemia, you would have the lovely, exciting task of peeing in a cup and analyzing your own urine. And actually, we have had one individual diagnose themselves with a UTI because he had everything in there that was not supposed to be in there. And he went to his doctor and he got some antibiotics and he was a lot happier and feeling a lot better. All right, podocytes. Fun things. The filtrate that makes it through the capillaries, the interstitial fluid, the podocytes, those filtration slits, is sometimes referred to as the ultrafiltrate. It's in the glomerulus, in the Bowman's capsule, I should say. It is ultrafiltrate, and I usually just say filtrate, but it could be classified as ultrafiltrate. It has not been touched by the processes of reabsorption or secretion. The substances, the fluid that is present in the glomerular or in the Bowman's capsule is identical to the plasma with some exceptions. The proteins that are present in the plasma, and in this little figure here, it's a pretty picture here. I'm going to highlight these in green. They're already green, so we're just going to help them out. These proteins that are found in the plasma remain in the plasma. So do the red blood cells and the white blood cells, as I previously mentioned. The substances that get into the ultrafiltrate, though, include water, of course, the electrolytes, urea. Okay, urea. Interesting molecule. Urea is a waste product of protein production, protein metabolism. It is kind of a cool little product. It's soluble in lipid membranes, so it can go in and out of these compartments with ease. And other types of molecules that you would expect to be found within the plasma, they're in the ultrafiltrate. Okay, they're in the ultrafiltrate. Importantly, and this is a really, really super important concept to understand, especially to understand a concept called renal clearance. Renal clearance refers to how quickly a substance is removed from the bloodstream and excreted in the urine by the renal system. A lot of drugs are metabolized in that way. They're cleared by the renal system. And so to understand renal clearance and to understand things like how glucose reabsorption occurs and how glucose receptors might be saturated, etc., we need to understand that if I have, as just an example, if I were to take a blood sample from my arterioles, so not necessarily right there, but take a blood sample and I were to measure, let's just say 100 milligrams per deciliter of glucose in my blood. Okay, then that means that I'm going to get 100 milligrams per deciliter of glucose in my ultrafiltrate. Okay, that's critical to get that, to understand that, to think of that, and be aware of that relationship in concentration.